Hi, everyone. I can see the attendee list is filling up. Yeah, so we'll try to start promptly. First of all, this is July 1st. Happy Canada Day. This is SIAM Life Sciences 2020 Plenary, one of several. The speaker is Jeremy Gunawardena, and before I introduce him, I'll tell you how this is going, how we're planning to make this work. So Jeremy is here. And the talk itself will be a video recording. So we'll hit play, and then we'll get to watch the talk. And then afterwards, because Jeremy's here, we can take questions. So for questions, what we'll do is that audience members can type the questions into the chat. And then after the talk, I will go through and read them and get responses from our speaker, right? So that's the plan. You can type the questions, attendees can type the questions anytime, and then at the end, we'll go address them in discussion. Okay, so it's my great privilege to introduce Jeremy. He's trained as a classical mathematician. His PhD is in algebraic topology from Cambridge. And after that, he has spent time at all kinds of different places, including University of Chicago, Stanford, MIT, ENS in Paris, also several years at Hewlett-Packard, HP Research, before finally landing at Harvard in what I believe became the Department of Systems Biology, where he is now, where he is appointed now. Now, at the very moment, he's actually in the UK, where he's joining us from. Right, so he has a lot of research, including, as of this morning, at least over 4,000 citations in original research, and many of us know him for the research, as well as several commentary articles that have influenced the systems biology community, and including one that many of you may have read, and if not, I encourage you to read it, a 2014 commentary in BMC Biology where he says, I quote, "Models in biology are accurate depictions of our pathetic thinking," which I think is a really great quote. And today he is going to tell us about Hopfield barriers, the interplay of mathematics and physics in molecular biology. Okay, so I will go ahead and initiate the video.
By saying thank you to the organizers, Alla and Angela, for inviting me to give this talk, and also for reorganizing everything in the light of the pandemic. Thank you also to John for his introduction. I'm very sorry not to be there in person, but in fact I'm even more remote than that. For reasons of bandwidth problems, we're taking the precaution of recording this. But I will be there for the broadcast and for the question and answer. So I hope if you have any questions, you will get in touch. Let me begin with the outline of what I'm going to discuss today. So I'm going to start by trying to set what I'll say in a somewhat broader context in respect to some of the other strategies that people have adopted for bringing mathematics and physics to bear on biology. I'm then going to turn to a particular strategy that we've been adopting, which centers around how energy is used in particularly the molecular realm in biology. And I'll then turn to the mathematical approach that we've taken, which we call the linear framework. It's based on, it's a graph theoretic way of thinking about Markov processes. And then I'll turn to three stories that come out of using the linear framework that tell us something about these issues of energy and the interplay between mathematics and physics. Okay, to get started, let's begin with what I call getting behind the data. So I think we can discern two strategies very broadly that are being used in the modern molecular biology, or what we call systems biology. One of them starts from experimental data and seeks statistical patterns. And the other starts from biological knowledge on the basis of which a mathematical model is constructed, which is used to account for the data and experiments. And I've picked out two representative papers here from many other interesting papers in the literature, merely to illustrate the two strategies. I would say the first strategy is perhaps the dominant one at the moment, and it reflects the confluence of two developments in biology. One is data, which is becoming available in ever increasing amounts. We're literally being drowned in it. Particularly sequencing data, but increasingly also imaging data, and now single molecule data. And that development has coincided with a development in computer science, which is the coming of age, if you like, of particular ways of analyzing data, machine learning in its various forms. And this confluence has become very exciting in biology and is driving a great deal of forward momentum in the subject. These are very productive, very influential strategies that have been taken. Stepping back a little bit, one of the common aspects of both strategies is that they both depend on the data in one form or another. And the data that we have is contingent on the particular experimental methods that we have at our disposal at this time. Those change over time. And whatever experimental methods we have, they always provide a sort of projection of what's really there, the underlying reality. And as a result, the stories that we end up telling can become biased in some ways by those particular contingent choices. So it raises the question of whether, in fact, as mathematicians and physicists coming to biology, there might be another strategy for approaching biology, and a strategy of getting behind the data to try and get to the fundamental reality that lies behind. Now, this is really a very old debate in science. You can trace it back to the beginnings of science in the 17th century, to the empirical perspective, perhaps stated very clearly by Francis Bacon, of starting with the data, in comparison to the rationalist or theoretical perspective that Descartes put forward of starting with theory. And this quotation from Albert Einstein, I think, in the modern era, sort of illustrates, I think, how profound this contrast can be. Einstein is saying in a letter to Werner Heisenberg that he had started off from thinking that you must begin with the observable quantities, but in fact had then come to change his mind. I think this was very much influenced by his work on Brownian motion, that it's the theory that decides what we can observe. And I think there's something very profound about that comment. And my mate, Rob Phillips, illustrated here, has said very much the same thing in a very nice article that appeared a few years ago, in which he contrasted, he was looking at sort of the typical interactions of theory and experiment, and the typical style of a paper is that one does a lot of experiments, these occupy figures 1 to 6, and at the end, a model is constructed, and this model is used to account for the data that's available, and that becomes figure 7. And he was saying, well, how about doing it the other way around? And is it not possible for theory to drive what we look for and the kinds of experiments that we might want to undertake, and become figure 1 theory rather than figure 7 theory? And I think this is very much in keeping with the idea of trying to get behind the data. Well, how do we do that? And I think there are many ways that we might think of doing that. I should say, perhaps in parentheses, that the idea of getting behind the data tends to, biologists, experimentalists tend to react with consternation to this, because if the history of biology teaches us many things, one of the things it teaches us is that ignoring the data and indulging in speculation can be extremely dangerous and take you very far from what's really going on. And the point here is not to ignore the data or forget it, rather to rely on it. It gives us our knowledge of what goes on in biology, but also to realize that we do have some fundamental understanding of how molecules come together. That comes from physics. And perhaps that provides an entry to trying to get at the reality that lies behind the particular projections that we get from experimental data. So following that thread, one of the things we've been very interested in is the idea that of looking at how energy is used in biology. Now, at one level, of course, this is very obvious. Life depends fundamentally on the expenditure of energy. We all know that, and it's been pointed out and described in detail by many people. I've just illustrated perhaps two of the well-known examples: Schrödinger's work on his book on what is life, and Ilya Prigoghine's and the work of his school in Brussels on dissipative structures in biology. So order and organization emerge in biology through because of the expenditure of energy. So all this is very well known. So it's in some ways surprising that despite that, very clear understanding, the functional implications of what energy buys you at the molecular level are still not well understood. And as an illustration of this, at the bottom of the diagram, I contrast, these are schematics of how genes are regulated. On the left, in the bacterial setting; on the right, in the eukaryotic setting. And this has been a problem that we've been very interested in the lab. And the contrast between these is rather dramatic. In the bacterial setting, the regulatory information is very close to where the gene itself exists, and the regulation happens through fairly straightforward protein-protein interactions. When we move to the eukaryotic setting, it's enormously more complicated, and there are two aspects. There's one is the information, the regulatory information is strewn across the genome in enhancers which might be very far from the transcription start site. There's a lot of geometry and topology involved in bringing those pieces of information together. But also there's an enormous amount of energy expenditure, and this is involved in organizing and reorganizing chromatin, in post-translationally modifying co-regulators, histone proteins, the transcriptional machinery, all of which is a form of energy expenditure. And these two aspects, the explosion of information and the energy expenditure, I think are very closely related. So we know all this. This is well understood at some level in terms of the molecular players, but the functional implications of this are not. And it seems to me that if we are to understand how genes work, particularly in higher eukaryotes, in animal cells, and particularly during the course of development in building the organism, where genes have to accomplish really amazing tasks, then we do need to understand how the expenditure of energy leads to functional capabilities which are not present in the bacterial setting. And despite that, by and large, the mathematical models we've typically used to study gene regulation have been based on equilibrium thermodynamic assumptions. So I think this is a place where I do believe that we have work to do to follow the energy. And that's what I hope to show you is some of the things that we've been doing in that regard. Okay, so let's move on. So I think the first person, I think, in the biology literature to really see with clarity the functional significance of energy was the physicist John Hopfield. And his work was very influential. If you talk to any molecular biologist and say kinetic proofreading, they'll say, oh yes, of course, kinetic proofreading. When I first came to biology, I was recommended to read this paper. It took me 10 years to understand it. Hopfield was interested in the problem of error correction during the production of RNA and DNA. About the time he was working in the 1970s, the first measurements of error rates in replication and transcription and translation were becoming available, and the numbers that were coming out were sort of one error in 10 to the 9 bases, 10 to the 10 bases, and these were extraordinary. And Hopfield was trying to understand how this was possible. The drawings at the bottom are slightly more formal versions of the drawings in Hopfield's paper, and he's formalizing this in thinking about a correct and an incorrect substrate being detected by some biochemical mechanism. And the first means of detection is binding, and typically discrimination takes place through off rates. The correct substrate sticks around for longer, it has a slower off rate. And the error rate for the discrimination process, which you can think of as the probability of the incorrect substrate being chosen compared to the probability of the correct substrate being chosen. I should say that this analysis is at steady state. So Hopfield's thinking about this mechanism as operating at steady state. So there's some error rate, let's call it epsilon, that just comes from binding and unbinding. And Hopfield's idea is very simple. He said, well, if you get an error rate of, say, epsilon with one binding, unbinding, why not take 2 bites of the cherry? And that's illustrated on the right. You allow for the possibility that there's one binding going from 0 to 2, or 2 bindings and going from 0 to 1, and then from 1 to 2. And if you allow 2 bites of the cherry and you get an error rate of epsilon in each bite, then you might expect to get an error rate of epsilon squared from 2 bites. So that's the basic idea, but there's a problem. And the problem, which of course Hopfield understood immediately, was that this doesn't work if the system is at thermodynamic equilibrium. And why it doesn't is a very important point, which is well understood by physicists, but perhaps not by us mathematicians so much. And as a pure mathematician by training, I was sort of amazed to discover, rather late, that there are 2 types of steady states as far as physicists are concerned. The height of the water in the sink can achieve a stationary state where it's not changing over time, either because there's no supply and no efflux, or because supply and efflux are balanced. And these are fundamentally physically different. In the situation on the left, this is a state of thermodynamic equilibrium. It's a state in which very, very strange things happen, strange in the sense that they're really quite astonishing. They go by the name of detail balance and microscopic reversibility. We'll say explicitly what these are in a little while. These properties don't hold away from thermodynamic equilibrium. A more familiar way of thinking about it is that if you take a movie of an equilibrium system at the molecular level, you can't tell whether it's running forwards or backwards, but away from equilibrium, there's an arrow of time. And more importantly, for what we're just talking about, the situation away from equilibrium is that the probabilities of states are dependent on the path taken to reach them, whereas at thermodynamic equilibrium, they are independent of the path. And that is the clue to why the previous scenario doesn't work at thermodynamic equilibrium, because if the system's at thermodynamic equilibrium, it can't tell whether it took 1 bite or 2. That's path independence. And the only way to break this is to introduce energy into the system, to break detail balance, to keep the system away from thermodynamic equilibrium. And at that point, the probability of being selected begins to depend on the path that is chosen. And if you are clever in examining the parametric dependencies, and that's really the essence, the hard part of what Hopfield did, was he found the parametric conditions under which there's a good balance between the two paths, and as a result, you get an improved error estimate. It's not as good as epsilon squared. It never can be as good as epsilon squared, but it can be better, a lot better than epsilon. And that's this idea of kinetic proofreading. And it's been influential in biology because most discrimination systems that anyone has looked at, I think, show this expenditure of energy at these discrimination steps. And kinetic proofreading is thought to play at least an important role, if not the full role, in the very high levels of discriminating capability that molecular systems have evolved. So that's kinetic proofreading, and that's typically what people remember when they talk about Hopfield. But I think what Hopfield was telling us was actually much more profound than just about discrimination. I think what he was saying was really a very fundamental principle that applies to any form of information processing. And so our modern take on what Hopfield is telling us is where this idea of a Hopfield barrier comes from. So suppose we have any information processing task. We looked at discrimination. It could be detection. It could be amplification. It could be sharpness, which we'll come to, or amplification. It's the same thing. It could be sorting, classification, learning. It doesn't matter. Whatever information processing task is being considered, if the machinery that's trying to do that is operating at thermodynamic equilibrium, there's a fundamental upper bound to how well it can perform that task, and that's set by detail balance. It's physics. You can't get away from it. And the only way to get past it is to expend energy, take the system away from equilibrium, and in that case, there's sort of a suggestion in Hopfield that if you expend enough energy, then you can make the error rate as low as you can. It's more subtle than that, and we'll come back to that later. But this is where this idea of a Hopfield barrier comes from. The Hopfield barrier is the limit to an information processing task that holds at thermodynamic equilibrium. And what Hopfield did was to identify that barrier for the case of discrimination. Now, once you get beyond the barrier, life gets much more complicated, because in Hopfield's very simple scheme, yes, you can keep the error rate very low, make it as low as you want, but the system gets slower and slower. So that's not good. And in real systems, you may have to trade off different kinds of functions that you want to achieve. So this is the starting point for a kind of third strategy that we might use to start thinking about how molecular systems work. Okay. So that's the Hopfield barrier. Now, in order to explore this further, we use a particular mathematical approach, which is called the linear framework. And one way to think about it is that it's taking the pictures that Hopfield wrote down in his paper, and I wrote down in a slightly more formal way on an earlier slide, and turning that into a full-blown graph-based approach. So, whoops, here we are. We start from a graph shown here. It's a directed graph in which the edges are labeled. The vertices of the graph you can think of as microstates. Shown here is the kind of graph we might use to study gene regulation. It contains binding states for a transcription factor, and it contains a binding site for RNA polymerase. And the edges, the directed edges, represent transitions between these microstates. And the labels, so the labels we think of essentially as rates. They have units of inverse time. And we think of them as basically the generators for infinitesimal generators for a Markov process. So a Markov process is described by a conditional probability distribution, and in the box at the top is the relationship between that conditional probability distribution and the infinitesimal rate, which is the label. Now, the label, and a couple of labels are shown here. Don't worry about the notation. You need a slightly elaborate notation to talk about graphs like this, but we're not going to get into that. I just want to draw your attention to the fact that the label can be a complex algebraic expression, because it describes the interaction between the graph and the environment in which the graph is placed. In this case, the environment contains two entities, a ligand L, and the concentration of the ligand, chemical potential, if you like, enters into the label for the on rate, as it were, for the transition, the binding transition. At the moment, it's a simplifying assumption. One can allow the assumption to be relaxed. We'll basically assume that whatever's in the environment, if it interacts with the graph, if it binds to vertices, that does not change its free concentration. So in the physics literature, this is equivalent to thinking of the pool of molecules like L as a reservoir. That's the common assumption in the physics context of depletion from the reservoir doesn't change its chemical potential. Okay, so with this data, we can describe a dynamics, and it's very simple dynamics. It's basically one-dimensional chemistry. We treat each directed edge as a chemical reaction under mass action kinetics with the label as the mass action rate constant. Now, if you follow that prescription and write down the resulting differential equations, it's clear that they must be linear because every edge has only one source vertex. And I've just given you a prescription for writing down a matrix equation shown in the bottom box. And the matrix in question is what graph theory is called the Laplacian matrix of the graph. Now, this Laplacian dynamics is precisely the master equation of the Markov process. Now, this relationship between the graph and the Markov process is one-to-one. If I start from a Markov process well behaved in the sense that it has infinitesimal rates, I can construct the graph, and the Laplacian dynamics on the graph is the master equation. So this is just another way of doing Markov processes. Interestingly, the graph doesn't make much of an appearance in the Markov process world, partly because I think in the present time, we've been mostly focused on steady states, and Markov process theorists are interested in many more things, dynamic things. The graph approach can be used in the dynamics as well. That's another story. Now, the other place where the graph makes its appearance is in the physics literature, and this goes back to work in the 1960s of Terrell Hill and in the 1970s of Jürgen Schnackenberg, who developed this as a way of looking at non-equilibrium physics. And their work was very significant, and we keep coming back to that to discover that they were there before us. But it sort of got lost from sight in the physics literature for reasons I don't understand, and is only recently resurfaced in what is called stochastic thermodynamics. So this approach really comes from biochemistry. That's our starting point, but it draws on these other disciplines to a great extent. So much of what I'm telling you, if you were a stochastic thermodynamicist or a Markov process theorist, you'd say, oh yes, we know all about that, but from a slightly different point of view. Okay, so what we're interested in the moment is steady states. And so we're going to look at the master equation when the left-hand side is 0. So we're looking at the kernel of the Laplacian. So I'm just going to run through some of the things that we know. And very quickly, so first of all, if G is strongly connected, which is the case for all the examples we'll look at, the kernel of the Laplacian is one-dimensional. If you're interested, just take a look at the references. It's quite straightforward. We can create a canonical basis element in the kernel of the Laplacian using the matrix tree theorem. And this version of the theorem goes back to Bill Tutty. What it says is that if I look at a particular vertex I, then I can construct the ith component of this basis element in the following way. I look at all spanning trees of the graph which are rooted at I. So I think everyone knows what this means. A tree is a subgraph which has no cycles if edge directions are ignored. It's spanning if it includes all vertices in the graph. and it's rooted at the vertex I if there are no outgoing edges from I. So we choose the spanning tree, we look at all the labels on the edges of the spanning tree, multiply them together, and we add those up over all spanning trees. It's a very simple formula, it's a very beautiful formula. It has enormous implications for what we're going to talk about, as we'll see. So this gives us a basis element, and once we have a basis element, it's very straightforward to eliminate the last degree of freedom by normalization and to obtain the steady state probability of vertex I, which now takes the form of a rational function of the edge labels. So these are the quantities that we're particularly interested in. And in the case of the particular graph for gene regulation that I showed you on the last slide, we typically look at the function in which we look at the probability of polymerase being bound. And we take that as proportional to the expression rate of the gene. So that's just one example of how we get a functional response from that particular graph. Okay, so we need a few bits and pieces. So what I've talked about is all mathematics. Where does the physics come in? And in particular, what does it mean to be at equilibrium and what are the consequences of that? So if the graph is at thermodynamic equilibrium, then two things have to be true. One is that it must be reversible. And what that means for us is that if there's an edge from I to J, there must be a reverse edge from J to I, which is physically the reverse process. Now, I should say that in the Markov process literature, reversible has a different meaning. It means what I'm about to say now, which is what the physicists call detail balance. And detail balance is this amazing condition that basically says that if I pick a pair of reversible edges between vertices I and J, those two vertices are basically in equilibrium with each other, irrespective of all the other connections in the graph. And that's represented by the formula that you see there. So it's as if at thermodynamic equilibrium, the system uncouples into pairs of vertices, which are linked just by the steady state probability distribution. So this is really amazing. And one of the interesting things about detail balance is that although it's a property of the state of thermodynamic equilibrium, it can't be deduced from thermodynamics. In fact, it relies on a much deeper property. It's the reversibility of the fundamental laws of physics, time reversibility of the laws of physics. So it's really quite a fundamental property. It has important consequences for us because if detail balance holds, then we can construct a different basis element in the kernel of the Laplacian. You take a path from a reference vertex, one, it can be chosen arbitrarily, to the vertex you're interested in, I, and you take the product of the label ratios along the path. And this gives you another basis vector in the kernel of the Laplacian. So as you see, this is a very, very different kind of expression. And from what I just said, captures the idea of path independence, because what detail balance tells you is that the number you get doesn't depend on the path you take. And that's the sense in which steady state probabilities are independent of path when you're at thermodynamic equilibrium. So this is what Hopfield ran into in doing this analysis of 2 bites of the cherry, but seen here in a formal way because we have the graph at our disposal to do it. You should think about that in contrast to the matrix tree theorem on the previous slide, where, as you can see, you can't just pick any old path. You have to worry about all the paths, all the spanning trees, and the MTT does the bookkeeping for you. Okay, so the contrast is really extremely marked and is going to be important for us. Okay, so a couple of further points. One is that detail balance is equivalent to a different condition, which is that if you take any cycle in the graph, the product of the labels going clockwise must equal the product of the labels going counterclockwise. This is easy to see, and its advantage is that it doesn't depend on the steady state probability distribution, it depends only on the labels. So this is the cycle condition. And that's also something quite useful to keep in mind when thinking about what happens at thermodynamic equilibrium. And finally, to make the physics connection explicit, physicists know that in an equilibrium context, there is a free energy landscape. And we can think of the graph, the vertices of the graph representing the minima of the free energy, and the ratio of the forward to the reverse transition rates between the two microstates or vertices is a function of the free energy difference. So this is a sort of basic insight of physics. And if you plug this insight into the prescription for calculating steady state probabilities, now using the equilibrium basis rather than the non-equilibrium basis vector, then what you get out of it is precisely equilibrium statistical mechanics. And you can identify the denominator of the rational function as being a partition function for the appropriate thermodynamic ensemble. So what this tells us is that this graph framework that we have here is a little garden in which we can do equilibrium statistical mechanics on the nose, but, and this is what's really important, you can also do non-equilibrium statistical mechanics. You have, in effect, a non-equilibrium partition function. Now, that's a big deal because, in general, non-equilibrium systems we don't have that capability in general for non-equilibrium systems. So this is one of the great advantages of the linear framework. Okay, so much for the framework, and let's now get on to thinking about what you can do with it. We all know about Michaelis-Menten formulas. Michaelis-Menten back in 1913 were looking at the reaction scheme that's shown on the top left. They were basically trying to understand how enzymes worked, and they went through a piece of algebra which torments all biochemistry students to derive their famous Michaelis-Menten formula shown underneath. And in fact, you can derive this very easily using what I've just told you about the linear framework. In fact, it's through enzyme kinetics that we really came to develop this graph framework, not really from physics. The graph in question is a graph on the enzyme states labeled as shown. And the essence of what Michaelis-Menten were doing was to make a steady state approximation that assumed that the enzyme states were at steady state. And if you use that and run with what we've just been talking about, it's very easy to derive the Michaelis-Menten formula from that graph. Now, one of the very interesting things that's been observed over many years is that this Michaelis-Menten formula crops up in an enormous number of other contexts, and the table on the right enumerates several of those contexts, the ones that have been known to us, where surprisingly, because they're much more complicated, the underlying systems are much more involved than the simple reaction mechanism, the Michaelis-Menten formula pops out. This has been something of a surprise to people in various areas. Some general statements have been made as to why it happens, but always with simplifying assumptions and typically with graphs when they're actually made explicit, which are very symmetric, grid graphs or arrays or things like that. So what the framework allows us to do is to actually make a much broader, much more general statement of what goes on here. So let's look first at the equilibrium setting. And very broadly, the Michaelis-Menten formula arises in a graph when the graph is a more detailed version of the very simple graph that works for the Michaelis-Menten reaction mechanism. And more specifically, let's make the assumption first that there's a particular ligand of interest whose concentration we're going to call X, and that the labels in the graph involve X as a monomial, so they're X to some power times some constant. And the constant could conceal all sorts of other things that are going on in terms of interactions with the environment. We're not worried about that. We're just looking at X. We need to be able to break the graph into two parts, V0 and V1 here, in such a way that the edges which go from V0 to V1 involve x to degree 1. So it's just linear in x. And all other vertices in the graph, x does not appear, it has degree 0. So if we can do this under conditions of thermodynamic equilibrium, then all of the vertices in V1 satisfy the Michaelis-Menten formula with respect to x. And conversely, namely, if you have a graph with monomial labels at equilibrium, in which a vertex satisfies the Michaelis-Menten formula, you can subdivide it in this way such that these degree conditions hold. So essentially at equilibrium we understand, and this is not difficult. It gets much more interesting away from equilibrium. Basically, provided you impose a very strong, the same result holds. The strong condition is that the x label, if it occurs on an edge, and you take that edge out of the graph, it disconnects the graph. It's no longer strongly connected. What that means is that the x labels, the edges on which there's an x label, occupy a tree within the graph, illustrated at the top graph that I've shown there. So this is a very strong constraint. And from a thermodynamic perspective, what it says is that x isn't responsible for any of the entropy production in the graph. Now, if you drop this condition of x acyclicity, as we call it, it gets much more complicated, and we don't have a full understanding of it. We have some results. For instance, the kind of sink structure that we see on the left, which I won't go into the details of, but there are also examples that don't fall under the scope of the results that we do have, such as the example on the right. And basically, we don't have a conjecture for what the complete statement is, particularly in the case where the graph is not reversible and they have edges which don't have reverses. So that's particularly difficult to tackle. So this is an interesting mathematical problem for which we don't have a full solution. But I should say that the results we do have include everything that's been proved in the literature, which has been shown in the literature in typically much greater generality. And just to emphasize that point, the splodges in these graphs can be arbitrary. So what goes on in those structures is completely irrelevant to the appearance of the Michaelis-Menten formula. So I would say that this is really illustrates for us why the graph is important, because it's a mathematical entity about which you can use to make these powerful statements about what's important and what isn't. And I think in the biological context, that's particularly valuable. Okay, so much for Michaelis-Menten responses. Let's turn to the Hill function, which, if you like, is a step up from the Michaelis-Menten hyperbolic response. So we all know that the Hill function is this family of simple rational functions. They were introduced by Archibald Vivian Hill, one of the founders of biophysics, in the early part of the 20th century, as a way of fitting data on hemoglobin. And Hill was very clear that this was basically an empirical fit. He gave a little kind of pseudo explanation for how it arises, which isn't biochemically plausible. But the Hill function has been extremely successful. It turns out to be really surprisingly good at fitting data. And at the bottom is an illustration from a problem which we got interested in. This is the expression of a gene hunchback in the early Drosophila embryo in response to bicoid, which was measured very carefully by a quartet of distinguished authors at Princeton, and they fit this to a Hill function with Hill coefficient 5. Now, one of the perennial issues in using Hill functions is that people keep thinking that the Hill function has some biophysical or biochemical meaning and try to interpret the Hill coefficient in those terms. Hill was very clear that this was not the case, and I've included a couple of the papers below, which out of many papers which keep reminding people that no, it's just an empirical fit. It doesn't have a biophysical or biochemical justification. However, the linear framework allows us to take another look at this. So what we're going to do is to look at input-output response functions for a single variable. So think about a graph. It has some distinguished ligand which is binding to it, that concentration of which is x. And we're looking at typically a steady state probability of some vertex or some linear combination of those. It doesn't really matter. So that we have a steady state response function. And we're going to look at the sharpness of these functions. And we do it by first normalizing to the half maximal value shown on the top right. And then having got that normalized function, we're going to estimate its sharpness with two quantities. It's very important that we use two here. One of which is what we call the steepness. It's the maximum of the derivative. And the other, which we unfortunately call position, it's a terrible name, is where that maximum occurs. So illustrated at the bottom there. So we're going to look at position steepness plots for these response functions. We have to choose one. So we're going to choose the one for the gene regulation graph that I showed you earlier, and the output function that I showed you earlier. I'm sorry, this is a little bit dense, but let's work through it. So the big plot on the right is illustrating, so think of it as randomly selecting parameter values, plotting position and steepness on this plot, and then determining the boundary of that region when the parameters lie within a certain range, which is stated at the top. Now, this particular plot is for 2 binding sites plus polymerase, and you might wonder why 2, and there's a story there, so we'll come to that. But what you can see is that if the graph is at equilibrium, the blue region, it has a shape which basically, although you can't see it, it has a cusp that lies on the magenta line. The magenta line, by the way, I should have said, is the locus of position steepness for the Hill function. And what's marked are the integer values of the Hill coefficient. So there are 2 sites, and the blue region has a cusp that's very close to, it lies on the Hill lines, very close to Hill coefficient 2. Now, this is generally true at equilibrium. And in the inset, what you see is an equilibrium plot, now for 6 sites plus polymerase. And what's being shown there is what changes when the parametric range increases. And what you can see, and I hope you can see because it's a little bit small, is that basically the boundary of the region stabilizes. There's an asymptotic boundary, and the boundary is reached pretty much when the parameters lie between 10 to the minus a and 10 to the a, where a is about 4.5 or 5. About that point, we can't get the boundaries to move beyond that, numerically speaking. So these equilibrium shapes have a very interesting geometry. And basically, what we see here is that the Hill function, whose coefficient is the number of sites, is a barrier. You can't get a response function to have a position and a steepness which are both higher than that of the Hill function. However, and coming back to the bigger plot, if you're now away from thermodynamic equilibrium, you can exceed this barrier. And in fact, in this case, for 2 sites plus polymerase, you get a similar cusp-like behavior on the Hill line, but now getting to Hill coefficient 4. So with 2 sites, you can get a Hill coefficient of 4, provided you're away from equilibrium, like nearly 4. So basically, this is the evidence for the result, which I'm putting in quotation marks for reasons I'll explain. So this is a general statement. If you have a response function on a single ligand, then basically the Hopfield barrier for its sharpness is the Hill function on the number of sites. So the Hill function, which has been for 100 years basically an empirical thing, is now revealed as actually having biophysical meaning. It's a Hopfield barrier. And the quotation marks are there because we don't have a proof of this. We have a lot of numerical evidence done from many different standpoints, which leaves us completely convinced that this is correct. But proving this seems quite hard. And I have to say, we haven't sort of rather given up trying, but partly for distraction with other things. I think there's something very attractive and interesting about what's going on here in the geometry of this map, which takes these rational functions to position and steepness, seems to have some very interesting geometry. Okay, so I'm showing below the statement of the theorem the actual rational functions, the highest order terms of these, and you can see that at the equilibrium situation, there's a relationship between the degree of the rational function and the position of the cusp. But when we get away from equilibrium, the situation changes completely. So for n equals, little n equals 2, capital N is 8 minus 2 is 6, but the cusp is at 4. So we don't even have a conjecture for what the relationship is between the degree and the cusp. So there's lots of interesting things here. Okay, so that's the Hopfield barrier for sharpness. And finally, let me come on to the last part, which has to do with these challenges away from equilibrium. So let's go back to the matrix tree theorem, which is really very interesting here. Let's think a little bit about its implications. So one of the things that is very easy to see is that if I have a vertex I and I pick any edge which is not outgoing from I, then that edge must be on some spanning tree rooted at I. And therefore its label is going to appear in the formula. So what that means is that pretty much every label in the graph is important in figuring out what the steady state probability of any vertex is. So this global parameter dependence is really very striking and really rather problematic. So one way to think about it is, imagine that we have chosen the rates, the labels, so that the system is at equilibrium. And that we have a very simple way of calculating things, just taking paths. But now we take one edge and we change one rate to be away from equilibrium. And all hell breaks loose. Steady state probabilities begin to depend on everything in the graph. So this is really very alarming and is an indication of how intransigent these steady state probabilities are. Now it's even worse. And as you can see, when you start asking yourself, how many spanning trees do I need to enumerate in order to exactly calculate the steady state probability? And the answer is way too many. And this is the reason that we were restricted in those plots to n equals 2, because away from, if you have n equals 2 transcription factor binding sites for this gene regulation graph, there are 384 spanning trees, but there's no way we can deal with what happens when we increase the number of sites by 1, as you can see from the number of spanning trees that you end up having to deal with. So this taught us a very interesting lesson, which is that the art of doing non-equilibrium physics, as I like to annoy my physics friends with, is the art of throwing away trees. They really are not conservationists in this respect. And Hopfield's paper is a beautiful illustration of exactly how to do that, because Hopfield doesn't go through the calculation in full. He doesn't write down all the spanning trees. He just brilliantly finds the parametric conditions which allow only one of the spanning trees to really dominate. And so this is the art of doing physics. But the problem with this is that biology may be operating in a very different regime where, in fact, things depend on lots of little things contributing to their effects. And we kind of see that in a lot of biological contexts. So this whole space where we can't find dominant trees has been dark. We haven't been able to get into it. And to me, this is one of the great challenges in the way of further progress in this area. So there is a little bit of hope, and this is very new work from our lab. I'm probably running a little bit close to time, so I'll go through this very quickly. What this is saying is that let's suppose we follow a stochastic trajectory of the Markov process, and we count the occurrences of a cycle, any cycle, one is shown below. So we follow the trajectory, and the cycle can start in any microstate vertex, but once it's started, you have to follow it exactly. There can be no excursions away from it. So we count only those full occurrences. And then we do the same for the reverse cycle. And we take the ratio of those two counts at a particular time t, and we let t go to infinity. That limit exists. And that number has a very interesting interpretation. As you can see, it's given by the ratio of the two terms in the cycle condition. The product of the labels going clockwise to the product of the labels going anti-clockwise. Now, this is very interesting because suppose the graph was at equilibrium and we went through this thought experiment of changing one rate so that it becomes away from equilibrium. Now, if that rate is not on the cycle that we're looking at, we could change multiple rates, but let's suppose that none of them are on the cycle that we're looking at, then the cycle condition doesn't change. And therefore that count has to be 1. However, if one of the edges on the cycle is a place where the rate has been perturbed away from its equilibrium value, then we will see that in this count. So what this count is telling us is that despite this global parameter dependence and this combinatorial explosion, this count is a local measure of whether or not energy is being expended on a cycle. That's actually quite nice because if you could actually observe the stochastic trajectory, then it's a way not only of knowing that your system is away from equilibrium, but also of saying which edges might be implicated in that. And that has a real bearing on the experimental challenges we face at the moment in trying to do some of these things. And I think we're still not there yet in being able to follow Markov processes at the molecular level, but single molecule techniques are really improving to the point we might hope that this might become possible. Okay, so I've tried to give you some hint of how the linear framework helps us to develop the strategy of following the energy. And let me leave you with a summary then of the things that I'm trying to say. I think Hopfield's old idea allows us to think about a sort of systematic way of trying to think, work our way back into molecular biology and interpret it from this thermodynamic and biophysical perspective. And that's something we're in the process of trying to do. One thing I touched on in the last couple of remarks about the cycle is that what happens away from equilibrium turns out to be much more nuanced than we had anticipated. As that cycle condition result suggests, it wasn't a statement about function, but once you start to look at function, it turns out that function is influenced not just by the fact that something's away from equilibrium, but where in respect of the graph is the energy going in. And if you put it in the wrong place, you might not get the functional capability you could if you put it in the right place. And even if you put a lot of energy in there, it may not be sufficient to achieve it. So the landscape of what happens away from equilibrium is very, very interesting and rich. And this is something that we're just starting to understand. Okay, so the challenges, I've talked about the mathematical ones, the experimental ones are very striking. I haven't really said anything about that side of things. And particularly detecting that we're away from equilibrium at the microscopic level is still very challenging. And then finally, something I haven't alluded to at all, but which is always behind the scenes, is the evolutionary question. And we know a lot about evolution and how it works, but actually I think the most puzzling thing is that it works at all. Because think of it like this. Think about the evolution of cars. If the evolution of cars happened in the same way as biology works, then we would see the Model T Ford turning into a Ferrari while always being on the road and capable of taking you from point A to point B. Clearly, that's not how it works in the engineering world, but that's how biology works. And that's really amazing. And I think if we are to really understand at the level of what happens at the molecules, then we have to get the foundations right. And this perhaps is helping us to put some of those pieces in place. So I will leave you with some of the people who've contributed to this, who have highlighted in magenta there my lab, and particularly my long-standing collaborator Angela De Pace in my department, and conversations with Angela have been very influential in leading us into the study of gene regulation. And on the right, a group of undergraduate students who've made particularly important contributions to the linear framework over the years. So thank you very much for your attention.
Okay. Thank you very much. Everyone, please join me in giving a round of applause with the round of applause button in Zoom. So we will now solicit questions. You may type the questions in the chat or in the QA feature, and then I will pass then I will read them out loud to Jeremy over the next few minutes. We have time to take a few. I saw there are a few comments during the talk about the slide resolution quality. I apologize for that. So part of it had to do with an aspect ratio issue. So there were black bars, and I was following along on another device, and it's true that some of the equations were like just barely legible. So I apologize for that. Okay. So while you're writing questions, maybe I'll ask one. Jeremy, are you still connected?
I'm sorry, June, I didn't catch that.
I'm just checking to see if you're still connected.
Yeah, I'm here.
Okay, while we're waiting to see if there are any questions, maybe I'll ask one. So, if I think about like larger scale biological systems, like for example, entire humans, if I asked all the humans, if I started singing the alphabet and I asked all the humans in the audience to raise their hand when I get to the letter J, let's say, and I made a dose response curve of that, letters of the alphabet versus when they raise their hand, it would be nearly perfect, right? It would be like slope infinity, Hill coefficient infinity. So what's happening there? Is it just that when you get to the organism level, we're just so wasteful of energy that we're nowhere near the Hopfield barrier?
So that's an interesting question. I guess I hadn't thought of it in those terms before. We know that at the organism level, we and other animals have rather remarkable capabilities. I mean, spotting faces, for instance, spotting a known face from a background of many faces is something that humans can do almost instantaneously, much faster than any machine that we've currently been able to make can do. Perhaps a way of looking at it is that for certain tasks which seem to be biologically important, we have very specialized hardware, which is perhaps highly efficient at those tasks. Whether that efficiency extends to energy issues is another matter. I mean, it's very hard to get an accounting in these terms when you get up to the organism level, because if you just think about an action potential on a neuron, which is a kind of very, very basic component of how cognition works at the organism level, well, you chew up 10 to the 8 ATPs to generate one of those action potentials, and that seems like an awful lot to be doing. But I think we don't understand the economics of running a large organism. I think perhaps it's better at the single cell level when we get into, say, phototransduction in a retinal neuron or something like that. It seems to be operating almost at the edge of what's physically plausible. Yeah, it's not an answer, but I think the economics is not well understood when we get up the scale.
Okay, thank you. Let's wait 30 more seconds to see if anybody is typing in. Okay, so if I understand correctly, the video recording will be available on the web, and in fact, if I understand correctly, that will be higher resolution than what Zoom is able to broadcast. So you'll be able to go back and look at the slide number with the fuzzy equation.
Oh, that's nice to know.
Jen, if I may jump in for a second.
Go ahead.
This is Ala Barsuk, one of the conference organizers. Before Jen wraps up and everybody starts to leave, I just want to remind you that tomorrow at the same time as we had this talk, we will have an early career prize ceremony, and the recipient of the prize will be giving a short talk. So you might want to come and hear about the achievements of the young person receiving this prize. And immediately after, it will be followed by the business meeting that will wrap up this conference. So if you want to participate in how this SIAM activity group is going to function and what's going to happen to future conferences and what we're going to learn from this one, then come and participate in that meeting as well. Thank you.
Okay, thank you, Ala, and thank you, Jeremy.
My pleasure.
Thank you, Jeremy.
Thanks, guys.